In this video, we're going to talk about monosaccharides. These are monomers that make up sugars. We are also going to have a look at how we can test for them. Monosaccharides are the monomers that make up carbohydrates. There are three main ones that you need to know about. They are fructose, galactose and glucose. Monosaccharides all share the same general formula, that is CH2ON, and you can use this to work out their molecular formula. They are mostly sweet tasting and they are soluble substances. So let's have a look at fructose first. Fructose is a hexose sugar with the molecular structure of C6H12O6. You just really need to be able to kind of recognise it. You don't need to be able to draw it or remember it in that sense. You just need to kind of be able to know that it's not glucose. So if we have a look at galactose, galactose is a hexose sugar as well, and it shares the same molecular structure. Now this means that these molecules are all structural isomers, okay? And again, you don't need to know this structure over here, you just need to be able to recognise that it's not glucose. Glucose. So glucose is a hexose sugar with the molecular structure again, C6H12O6, and there are two versions that you need to be able to know, alpha and beta glucose. Now, you do need to be able to draw out this structure. Okay, so this is one that you really do need to remember, and we're going to have a look at alpha and beta glucose in a little bit more detail. Let's take a look at alpha and beta glucose in a little bit more detail. This here is your alpha glucose molecule, okay? You've got your OH at the bottom and your H at the top, and I like to try and think of this in a triangle shape, so with an OH here, and OH at the bottom, okay, forming like an A shape to help me remember for alpha. If I flip this over to beta, you'll notice that the OH is at the top and the H is at the bottom, okay? So these are structural isomers of each other. Now, you do need to be able to draw these molecules, but luckily you can draw it in a slightly more simple way. So let's have a look. We'll flip back to alpha glucose because that's what we're going to draw. So if we start out with our simple hexagonal shape, okay, like this, you can then add on your OH and your H's at the sides. Okay, so in this case, we're going to have H at the top, OH down here, and the same on the other side. You've got H at the top and OH down here. Now, it's really important, okay, to make sure that it's your O that is joining up and not your H, okay, because it's the O that's attached to what would be your C, okay? And then finally, you add in your line here to represent your group at the top. Now, if you were going to draw your beta glucose, obviously you would just draw these the other way around, okay? So nice and straightforward. You don't have to add in all your other OHs and Hs, okay? Just make sure that you do add in this extra line here to represent that group at the top, and that is your alpha and your beta glucose. These simple sugars that we've been looking at are known as reducing sugars. This is because they are capable of transferring electrons to other compounds in a process called reduction. Now, we can test for the presence of reducing sugars in food substances, for example, by using the Benedict's test. So if we go over to the lab now, I will show you how it's done. Right, now that we're in the lab, we're going to have a look at how the Benedict's test work. So we've got some solutions here that we're going to have a look at, and they are made up of, we have some biscuit to test, we have some apple to test, and we are going to test some glucose and some sucrose in the hope that we can show you a very positive result and a very negative result. So these need to be in solution. So I've already put them here into solution for you. So we've got our apple in solution, we have our biscuit in solution, and then obviously our glucose and our sucrose are already in solution. So we're going to be using our Benedict's test, which uses our Benedict's solution. Now this is corrosive, so I am going to put on some goggles before we get started. We'll start with the apple. So we're going to take some of our Benedict's solution. Now we take equal parts, that's what we say. We take equal parts of our Benedict's solution. We're going to add it into our apple, like so. And then we're going to give it a little shake around so that it's fully nice and mixed. Next up we have our biscuit. I'm actually going to give this one a bit of a mix first, just so it's nicely mixed into that water. We're going to add, again, we're going to add equal parts Benedict's solution, give that a little shake. And then finally, we've got our glucose and our sucrose solutions. Again, we're going to do equal parts to both of those. Give them a little bit of a mix. Okay, so this is the test for reducing sugars. Okay, so apple, we've got biscuit, we've got glucose and sucrose. Remember, we're expecting a positive result from this glucose solution because we know that is a reducing sugar, and we're expecting a negative result from this sucrose solution because we know that's a non-reducing sugar. Okay, the main part about Benedict's is we have to heat it. So we add Benedict's and we heat. That's always what they want you to write about in the exam. So we're going to add these to some recently boiled hot water, and we're going to pop them onto our Bunsen burner over here, just to keep them hot while we time. Now we're going to time these for 5 minutes. So I'm going to start my stopwatch, which is over here. And we're going to make sure that we wait for a full 5 minutes and then we'll check the results. So now that we have reached our 5 minutes, we're going to have a look at the results. First up, we're going to have a look at the sucrose. So sucrose has stayed this lovely blue colour. Okay, so there's been no colour change at all. That suggests that we have a negative result, which we would expect because sucrose is not a reducing sugar. Okay, now we have our 2 food samples that we're going to have a look at. So we've got our biscuit here. So this has changed to a slightly green colour. That suggests that there's a little bit of sugar present. Okay, and the Benedict's test works as a scale in colours. It's a semi-quantitative test, and the colours show us how much sugar is present. Second food sample we have is apple. So I'm using this piece of paper here so that you can clearly see the differences in the colours. This has gone brick red. Now brick red is highest on the scale. This shows us that we have a lot of reducing sugar present. So there's a lot of sugar in that apple. Okay. And our glucose solution, which we know definitely is a reducing sugar, so this was to show us a definite positive result, has also gone that brick red colour. So those are our results at the front here. Now, you can see that there's a difference, especially between these two here. Okay, we can see there's a different level of sugar. However, these two are both brick red. There wouldn't be a way just from looking at them that you could tell which has more of reducing sugar present. So a really common exam question is for them to ask how you might gauge the difference. So you could take out the precipitate, you could dry it, and then you could weigh it, and that would give you a more quantitative way of telling the difference, especially if they've got all the way to this brick red colour along this scale. So we start off with our negative result, so we've got this blue colour, and then we go all the way along through our green, there might possibly be a yellow option in there, right through to our brick red colours there. And that is our Benedict's test. Now that we've been in the lab, let's have a look at the results. The Benedict's test is qualitative, okay? This suggests that the results only really give us an indication to the amount of sugar present, and there's no numerical data that we can analyse. So we started with a negative result, okay? The colour stayed blue, there was no change. Then we progressed, okay, through these colours until we reached brick red, and that was a result showing us the most sugar was present. The test is also subjective, okay, so it relies on somebody's opinion, okay? So my opinion on whether it was this colour or this colour would be slightly different to yours, okay? It just shows us that there's a range, okay, of more or less sugar. Now we could compare these to some known concentrations of glucose. That would give us a better indication of the glucose present. We could use a colorimeter. Again, that would give us some actual data. Or we could take out the precipitate, we could dry it and weigh it, and again, that would give us some more numerical data, giving us a better indication of how much sugar was actually present in these samples, okay? So the Benedict's test is mostly qualitative, it's subjective, but we could give ourselves some data if we looked at the colorimeter, dried the precipitate, or compared against some known glucose concentrations. In this video, we covered the structure of monosaccharides, the differences between alpha and beta glucose, and the Benedict's test. In the next video, we are going to cover the structure of some disaccharides.